In this video, let us learn about the sodium-potassium pump. Now consider the cell. The concentration of sodium ions is much higher outside the cell as compared to inside the cell, and the concentration of potassium ion is much higher inside the cell than outside the cell. Now if you look at the numbers, the concentration of sodium, like you can see in the extracellular fluid, is around 140 milliequivalents per liter, whereas in the intracellular fluid it is around 10 milliequivalents per liter. And the concentration of potassium in the intracellular fluid is about 140 milliequivalents per liter, whereas in the extracellular fluid it is just about 4 milliequivalents per liter. Now this concentration difference has to be maintained for a lot of vital functions of the body, a few of which will be discussed in this video later. Now, what helps us maintain this concentration difference? Like we all know, simple diffusion cannot maintain this concentration difference because what happens in simple diffusion is ions move from an area of higher concentration to an area of lower concentration till the concentration becomes equal on both the sides, right? So the concentration difference cannot be maintained. Then what is it that helps us maintain this concentration difference? It is a primary active transport mechanism, right? What is this primary active transport mechanism? It is the sodium-potassium pump, right? What sodium-potassium pump does here is it moves sodium and potassium from an area of low concentration to an area of high concentration, right? That is, it is moving against the concentration gradient, and if it has to move, the ions have to move against the concentration gradient, then it requires energy. That is why we call sodium potassium pump an example of active transport mechanism, because here the ions move against the concentration gradient with the use of energy, and this energy is utilized in the form of ATP molecules, which is broken down into ADP and inorganic phosphate, right? That is why we call it primary active transport, because the energy is directly obtained by breaking down of ATP molecule, and the pump itself has ATPase activity. Therefore, the pump is also called sodium potassium ATPase. Now, let's see what exactly happens here. Let's suppose this is a cell membrane, this is the extracellular part, and this is the intracellular part. So what happens here is 3 sodium ions move from inside to outside the cell, while 2 potassium ions move from outside to inside, both the ions moving against the concentration gradient with the use of an ATP molecule, right? So if you put it here, then in each cycle 3 sodium ions move outside, 2 potassium ions move inside. So like you can see here, for each cycle there is a net movement of 1 positive ion that happens from inside to outside. So the outside of the cell becomes more positive as compared to inside, which means that there is an electrical gradient that is generated, right? Because the pump creates an electrical gradient, the pump is also called an electrogenic pump. So, our sodium potassium pump is an example of primary active transport, which is also electrogenic. That is, it creates an electrical potential, and this potential is important for generation of resting membrane potential and for transmission of impulses. Transmission of impulses, whether in muscles or nerves, okay, everywhere it helps. Alright, let's look at another function. Suppose this is our cell. Now, we all know that the cells have a lot of proteins and organic molecules, and these molecules are negatively charged, large molecules. So these molecules tend to remain inside the cell, they do not move outside. So because they're negatively charged, what they'll do is they'll attract positive charges, right? Negative charges attract positive charges. So they'll attract positively charged molecules. Now, suppose these molecules move inside. They'll carry water along with them, right? These molecules will also carry water along with them. Now, suppose, imagine a situation where these positive charges enter the cell, positively charged molecules that is enter the cell. Now water molecules also enter the cell, right? So what will happen eventually is that these molecules will cause the cell to swell up, right? With all these molecules inside, it will swell up and eventually what will happen is it will burst. So our pump, by pumping out more positive charges outside, keeps a check on the volume. So sodium-potassium pump maintains the cell volume, right? Okay. The sodium-potassium pump maintains a high concentration of sodium outside the cell as compared to inside the cell. So these ions have a tendency to move inside the cell along the concentration gradient. There are other proteins and channels that are present in the cell wall that will help in movement of the sodium ions along the concentration gradient. So while they move, they'll carry with them other molecules. They can pull in other molecules along with them. These molecules, they are basically utilizing the concentration gradient as an energy source, right? For these molecules, the energy source is the concentration gradient of sodium ions, right? Therefore, this mechanism is called secondary active transport because the energy is not directly obtained by ATP. Okay, here the source is the concentration gradient. Therefore, it is called secondary active transport. Now, based on the direction of movement of these ions, it can either be symport or it can be antiport, right? Symport is when the molecules move along the direction of sodium, that is along the direction of basically the energy source, the concentration gradient. Example being glucose. And antiport is when the movement of the other molecule happens in the opposite direction. Example is calcium ions. Calcium and sodium move in the opposite direction in the cardiac cells, whereas glucose and sodium they move in the same direction. You must have heard of SGLT transporters. So that is what happens here. So this is another important function of the pump. Now let us have a look at the structure of the pump. Our sodium potassium pump has two important subunits, the alpha subunit, which is the larger subunit, and the beta subunit, which is the smaller subunit. The alpha subunit is more important here because it has the sodium binding sites on the inside, potassium binding site on the outside, and the ATPase activity near the sodium binding site to the inside aspect of the cell. Now the function of the beta subunit, which is the smaller subunit, is folding, targeting and anchoring of the molecule or the carrier protein to the cell membrane. Right. Now literature also mentions about a third unit which belongs to the FXYD protein family. FXYD protein family, which is tissue specific. Right? Now, what exactly happens during each cycle? Here, let me make it clear, this is the intracellular aspect, this is the extracellular aspect, and this is our pump. So, initially, the pump is present in such a way that the mouth is open on the inside aspect, right? And it is closed on the outside aspect. This is how the pump is present. Now, what happens here is, first, an ATP molecule attaches itself to the pump. This form of pump has high affinity for sodium ions. So, our sodium ions will come and bind to the specific binding sites that are present on the inside aspect because the mouth is open on the inside at this point, right? Once the sodium ions bind, the mouth closes, right? The mouth closes and these sodium ions are now trapped inside. At this point, what happens is this ATP molecule is broken down into ADP and inorganic phosphate by ATPase, which is present in the pump itself. So, this ATP is broken down and this phosphate group attaches itself to the pump. So, our pump becomes phosphorylated. The phosphate group attaches to the pump, phosphorylating the pump. Once the pump gets phosphorylated, it changes its conformation such a way that the pump is now open on the outside and closed on the inside, right? And the phosphorylated pump has less affinity for sodium. So these sodium ions are now released to the outside because the mouth is now open on the outside aspect and the phosphorylated pump has less affinity for sodium. Next, what happens is, even though the pump has low affinity for sodium in this form, it has high affinity for potassium. So two potassium ions will now come and binding to the specific binding sites on the outside aspect, right? Once it binds, the pump again closes its mouth, trapping the potassium ions inside. At this point, our pump is still phosphorylated. Now what happens is, the pump loses the phosphate group, becomes dephosphorylated at this point. Dephosphorylated form of the pump has less affinity for potassium, releasing the potassium ions on the inside, right? The inside mouth is open, outside is closed, right? This is one complete cycle where 3 sodium ions move outside and 2 potassium ions move inside. Let me repeat it once more. Initially what happens is, the pump is open, the mouth of the pump is open on the inside and one ATP molecule is attached. This form of the pump has more affinity for sodium ions. So the sodium ions will now bind to the pump. It will then close its mouth internally. It becomes internally occluded and 3 sodium ions are now trapped inside the pump. At this point, ATP is broken down and the phosphate group attaches to the pump, phosphorylating the pump. Now the phosphorylated pump has another conformation, that is, the mouth becomes now open on the outside. Mouth opens on the outside and phosphorylated pump has less affinity for sodium, so sodium ions are released. But the phosphorylated pump has high affinity for potassium, so the potassium will now come and bind to its specific binding site. After it binds, the mouth closes externally. So now it is externally occluded and the potassium ions are trapped inside. Once it gets trapped, the pump loses its phosphate group and it becomes dephosphorylated. Dephosphorylated pump has less affinity for potassium and these potassium ions are released outside. Right? This completes one cycle. Now, to conclude, what we saw in this presentation is that the sodium-potassium pump is an example of a primary active transport. Active because it utilizes energy, and primary because the energy is obtained by hydrolysis of ATP directly. It moves 3 sodium ions from inside to outside and 2 potassium ions from outside to inside the cell, thus maintaining a high concentration of sodium outside the cell and a high concentration of potassium within the cell. And it plays an important role in signal transmission, RMP generation, maintenance of cell volume, secondary active transport, which is important in a lot of tissues in the body. So that's it about the sodium-potassium pump.